All right, continuation of the male video. Now we're talking about the physiology of the system, or really how it works. So first and foremost, it's how do we make those male gametes? How do we make those 23 chromosome non-identical Darter cells that are going to be able to reproduce, fertilize, and create the zygote? This process is spermatogenesis. If you have any knowledge of Latin, genesis is essentially the creation of something. Sperm, as we're talking about it, the creation of sperm. This is the process which male gametes or sperm cells develop, and they are regulated by hormones from the hypothalamus and the anterior pituitary glands. So spermatogenesis kicks on when puberty happens and will continue throughout lifespan until and I was thinking about it, and quite frankly, sperm will decrease in its potency and effectiveness, but there is really no hard, strong cutoff when the system's just like, nope, we're done, shut it off. This is why sometimes you see very elderly man still be able to produce children. Now, it occurs in the seminiferous tubules by sperm forming spermatogenic cells. And here is the process of it. So you can see up here you have mitosis of the spermagonia. These are cells that will eventually differentiate into spermatocytes, which are going to undergo meiosis. Remember, all somatic cells can do mitosis. That is how we can produce and continue to produce gametes. So you have these spermatogonia, they undergo mitosis, puberty kicks on, cool. We've just unlocked meiosis. You are then going to take that one primary spermatocyte, it undergoes meiosis 1, produces 2 haploid secondary spermatocytes. Those 2 secondary spermatocytes undergo meiosis 2, and then we have a total of 4 spermatids. Those have 23 chromosomes, and these are going to eventually turn into sperm. The spermatids migrate into the epididymis to continue to undergo spermiogenesis when they eventually mature into fully mature sperm cells. And that is the process, very briefly, very skimming, of spermatogenesis. Now, you have sustacular cells. Sertoli are the nurse cells. These surround the spermatogenic cells to provide support. This microscopic anatomical arrangement forms the blood-testis barrier. There are several different barriers. I'm sure the most common one that you've heard of is the blood-brain barrier. There's the blood-placental barrier, and there is the blood-testis barrier, are some of the more common ones that are spoken about. This is essentially talking about how you have blood and then something. There is an extra layer of protection there. You see it in the brain. You see it when you have the fetus between blood placenta barrier, and then you see it at the testis too, because the body understands how important it is to preserve the reproductive system. Because remember, if you can't reproduce, you can't propagate the species, and your species is going to go extinct. Now, spermiogenesis. This is the process of sperm maturation. Spermatogenesis is the process of making sperm. Spermiogenesis is this process of sperm maturing, going from that spermatid to that mature sperm cell. Spermatids develop a head, a midpiece, and a tail as they become sperm cells. The head contains the nucleus, acrosome, and very little cytoplasm. This is going to be the thing that is going to have those 23 chromosomes, and it's essentially the most important part, the head. The midpiece contains mitochondria that generate ATP to power the flagellum, and the tail, that's mostly the flagellum. So if we think about it, The head is essentially just a nucleus full of DNA searching for that other gamete, that female gamete, so that they can fertilize and produce a zygote. The mid piece is essentially the housing of all the mitochondria that it's going to take up that fructose and that citrate to make ATP to power that tail, or the flagellum, to move that sperm through the female reproductive tract. Hormone control of male reproduction. Testosterone. It's the main hormone involved in the regulation of spermatogenesis and male reproductive physiology. It's regulated by a multi-tiered negative feedback loop, and we'll actually have a picture on it. We have the hypothalamic pituitary gonadal axis. This is going to regulate the hormones involved with testosterone production and testicular function. Pause for a second. What cell type produces testosterone? Hopefully, you said Leydig cells. Now, there is a chart here, which we're going to talk about and go through, which I'm going to do that first, and then I'm actually going to go up because I like pictures a little bit more. You have your first tier control. The hypothalamus releases GnRH, which is gonadotropin releasing hormone. This GnRH will stimulate the anterior pituitary gland to release luteinizing hormone or follicle stimulating hormone, LH and FSH respectively. Now, LH will stimulate testosterone production from the interstitial cells, Leydig cells. So if we see here, LH is going into the testis, FSH also going it. LH is going to work on the interstitial cells, and that's going to produce testosterone, where FSH, or follicle stimulating hormone is going to produce ABP, which stands for androgen binding protein. This is so that the testosterone won't essentially leave the testis because it is essential for the process that's going to be going on. Now, we have ABP binds testosterone. This testosterone is going to stimulate spermatogenesis and the development of male characteristics. And this negative feedback loop goes up here as levels of testosterone and inhibin rises. The secretion of GnRH, FSH, and luteinizing hormone decreases. So you can see here that once testosterone is made from luteinizing hormone at the interstitial cells, we have testosterone. FSH, follicle stimulating hormone, is going to talk to the sustentacular cells, produce ABP, androgen binding protein, and inhibin. When you have high levels of testosterone and high levels of inhibin, it's a negative feedback loop telling the hypothalamus and the anterior pituitary gland, hey, cut it out. We don't need any more. We got enough sperm. We got enough testosterone. Shut the system down. Now, this is just more me because I did my PhD on this system, or on the HPG axis, but what isn't listed here and everything is that your brain doesn't really listen to testosterone in this process. You actually have an enzyme called aromatase B, which will take testosterone, convert it into estrogen, and that's actually what shuts down this system. Just lore and trivia and knowledge because it's super cool. Now, let's go back to the actual written out version of this. So, testosterone is the main hormone involved in the regulation of spermatogenesis. That is the production of sperm. It is regulated by a negative feedback loop. HPG axis will regulate. We have our first tier of control, essentially the thing that starts the process, gonadotropin releasing hormone, GnRH, secreted by the hypothalamus. We have then the next step, second tier controlled. The anterior pituitary gland detects that GnRH and stimulates the secretion of follicle stimulating hormone, FSH, or luteinizing hormone, LH. Third tier control in the testes. So if you can think about it, I like to think about it like this. HPG, first tier, hypothalamus. Second tier, pituitary. Third tier, gonad. And since it's in men, it's the testis. Luteinizing hormone stimulates interstitial cells to produce testosterone. Follicle-stimulating hormone stimulates sustentacular cells to produce androgen-binding protein, ABP. This keeps the testosterone localized. And inhibin, which helps on the negative feedback loop to turn it off. Now, we have made sperm and we've matured the sperm. Let's use it. The process of essentially getting the sperm out of the body is ejaculation. It expels the semen from the penis under sympathetic nervous system control, and it occurs in 2 stages. You have emission and expulsion. Emission is essentially the movement through the system. So if we go all the way back into the anatomy section, emission is going to be moving it through. And then we have expulsion. This is when they accumulate in the urethra with ejaculation it being leaving. Now, testosterone levels increase dramatically at puberty, and this leads to the following changes. Puberty in boys begin at 12 to 14 years of age. The hypothalamus becomes less sensitive to inhibition, and we're starting now to ramp up production of GnRH, which then stimulates the activation of the HPG axis. And then we get those secondary male characteristics. We get the trigger of spermatogenesis, where we're starting to make sperm. And then once this begins, males are essentially capable of sexual reproduction. We will be going into puberty much deeper in a separate section, but this is just to get you started. I hope you guys enjoyed the male section, because the female section is a lot more complex and truthfully a lot more interesting. See you there.